Good day, students. So our subject is histology. So for our first lecture, we will only be talking about the introduction to our subject. Let us first define what is our subject. If you're going to ask someone what is histology, you would expect that that person will most likely answer you back, study of tissues. But what I want everybody to understand, that is how an elementary student would also define histology. But since you're already in college and you're supposed to, take up medicine in the future, you're supposed to, widen your definition of what is histology. I want everybody to understand, in histology, you do not just simply study the tissues and identify them. Okay? You also have to know their functions, and you also have to know how these tissues are arranged for them to form an organ. Okay? And for this one to make sense, I will explain this one later on in the succeeding slides. Since our subject is histology and histology is the study of tissues, we should also define what is a tissue. Most of the time, we define tissue as the group of cells. I want everybody to take note that there is more to a tissue than just being a group of cells. What do I mean by that? I want you to imagine, I will get a neuron from my brain, a hepatocyte from my liver, and a keratinocyte from my skin. If I will place them in one container, and since they already grouped together, I still cannot call them as a tissue. Why? A tissue is a group of cells with interrelated function. I want everybody to understand, the cells in the tissue need not to be of the same type of cell. Okay? The most important is their functions must be interrelated with each other. So again, a tissue is a group of cells with interrelated function. That's why I intentionally emphasize in this PowerPoint slide that the cells need not to be of the same type. They don't have to be of the same shape, same morphology. As long as their functions are interrelated, then they can make up a tissue. So this is a good representative photo of the nervous tissue. Since in the first part of the discussion in histology, we will first start with the basics. So we will first discuss the cell. So most of you would wonder, why study the cell in a subject that is supposed to study the tissues? Because for me, since tissues are made up of cells, you can never understand the characteristic of a tissue without understanding the characteristics of its individual components, which are the cells. You have learned from your anatomy and other science subjects that the smallest or the basic structural and functional unit of the human body is actually the cell. Okay? And I want everybody to recall, if this cell will be grouped with other cells, and as long as their functions are interrelated, they can form a tissue. Okay? And if tissues are grouped together and their functions are also interrelated, they can make up an organ. And an organ will always belong to a certain human body system. So in this picture, we're actually looking at the urinary system. So to best explain the organization in the human body, I want everybody to take a look at the urinary bladder. It's actually one of the organs in the body, okay? And then this urinary bladder belongs to the urinary system. Since the urinary bladder is an organ, okay, and organs are supposed to be made up of tissue, so we'll try to prove that one on this PowerPoint slide. Okay, so what we will do is, we will make a thin section, okay, of the urinary bladder, and place that thin section on a slide and apply stain, and we will view it under the microscope. And this is what we will see under the microscope. Okay, I want you all to take a look at the first X that would appear on the PowerPoint slide. That is actually, the X is actually positioned on one of the tissues, okay, that you can find in the urinary bladder. Okay, what do you notice? That area where you can find the X, okay, the X sign is actually composed of group of cells, because tissues are actually group of cells. Okay, the second X, okay, appears on another area where you can find another tissue. And what do you notice? That area is also made up of group of cells. Okay, so therefore, this PowerPoint slide will actually prove to you that the urinary bladder, since it is an organ, it's made up of different types of tissues. So what we will try to do on this PowerPoint slide is to prove that the tissues are made up of group of cells. Okay, the area that is encircled, okay, in the photo is actually one of the tissues that makes up the urinary bladder, right? Okay, and if we're going to shift the microscope to a higher magnification, that area will actually appear like this, okay? What do you notice? That particular area is actually composed of group of cells. So therefore, we have proven, okay, that tissues are really made up of group of cells with interrelated function. And if these tissues are grouped together, okay, they can form an organ. I want you all to take note that our subject is also referred to as microscopic anatomy, okay? Because if you can remember in your anatomy, you've been labeling structures that you can see with your naked eye. In histology, you will also be labeling structures, but this time, you need the help of a microscope. That's why our subject is always referred to as the microscopic anatomy. Okay? Again, let me emphasize that our subject is the study of tissues, but aside from identifying these tissues, you need to know their functions, okay? And you also need to know how these tissues are arranged to form an organ. And to prove my point, I want you to move to the next slide. So you have learned from your anatomy that there are four basic types of tissues. Okay, you have the epithelium, you have the connective tissue, you have the muscle, and you have the nervous tissue. What do you notice? All of these types of tissues have group of cells. Okay, and if you're going to combine these four basic types of tissues, then you can form actually an organ. Shown in the photo is the stomach, and stomach is one of the organs in the body. And since you've learned that for an organ to be formed, there must be a group of tissues, okay, with interrelated function. And I want you all to recall that in histology, it's not enough that you can identify the tissues. It's also very important that you know how these tissues are arranged to form an organ. So I want everybody to focus on the PPT, okay? The tissue that is situated nearest to the lumen, okay, of the stomach is actually the epithelium, okay? Below the epithelium, you have the connective tissue. And then below the connective tissue, you have the muscle tissue. And somehow distributed between the connective and muscle tissue is the nervous tissue. As you can see, these four, okay, were all grouped together to form an organ. And in this case, it's the stomach. I hope you can still remember from your anatomy that the lining epithelium functions to provide covering or protection to the organ, aside from the fact that some of them would actually participate in absorption and secretion. And the best example I can give you are the lining epithelia of our small and large intestine, because that is where we absorb the nutrients from the foods that we have ingested. Okay. I want you all to take a look at the photo, okay? You can appreciate from the photo that the lining epithelium of the stomach is situated nearest to its lumen, okay? So there must be a reason why the lining epithelium is positioned nearest to the lumen, okay? So that is now where the role of the histology will come into play. We'll try to figure out why the epithelium, okay, is placed nearest to the lumen of the stomach and not the connective tissue. I hope you can still remember from your anatomy that the stomach functions to produce hydrochloric acid. And when you say acid, it has something to do with being destructive. Okay? And among the four basic types of tissue, it is the epithelium that can actually provide protection. So, to protect the stomach from its own secretion, okay, you better place a tissue that functions for protection nearest to its lumen, because that is where you can find the hydrochloric acid. Okay, I want you to take a look at the picture of the epithelial tissue. Okay, can you appreciate that there are group of cells in there, because tissues are made up of group of cells? But what I want you to take note, the cells that make up the epithelium are tightly packed, meaning they do not tolerate spaces between them. Okay, so therefore you can also assume, okay, that the lining epithelium doesn't have its own blood supply, because if there's a presence of blood vessel between these cells, then these cells are no longer tightly packed and they cannot function for protection. I hope you can appreciate ba na that the cells have to be tightly packed so that the acid cannot go through between them and destroy the other tissues of the stomach. Okay, so can we all assume or no conclude, okay, that the epithelium doesn't have its own blood supply? Okay, primarily because of what? The cells in the epithelium are tightly packed together. Okay, so this is how the stomach would actually look like under the microscope. Okay, enclosed in the box is actually the lining epithelium of the stomach, and I want you all to take a look at the lining epithelium. The cells there are tightly packed together, so therefore there is no space for blood vessels. So therefore we have characterized the epithelium as avascular, meaning it doesn't have its own blood supply. So come to think of it, if the cells of the epithelium, okay, are not getting oxygen, then most likely these cells would die. Okay, so how do you think the organs in the body maintain the cells of the lining epithelium? Okay, so let's go back with our definition of histology. Histology is the study of tissues and how these tissues are arranged to form an organ. I hope you can still remember that. Okay, so you can still recall that the tissue situated nearest to the lumen of the stomach is the epithelium. Okay, and this epithelium doesn't have its own blood supply, but it's situated there because it's the epithelium that can actually provide protection against the hydrochloric acid in the stomach. Okay, I hope you can still recall from the previous slide that what you can see immediately below the epithelium is the connective tissue. Okay, and why do you think the connective tissue is situated immediately below the epithelium? Okay, connective tissue is highly vascularized, meaning the connective tissue has its own blood supply. So, the epithelium is actually there to protect the connective tissue from the hydrochloric acid, and what will keep the cells of the epithelium alive, because they will be getting blood supply every now and then, is the connective tissue. So you see, okay, so in histology we need to also study how the tissues are arranged to form an organ. You place the epithelium nearest to the lumen so that acid cannot destroy the organ, but you have to place connective tissue immediately below the epithelium because the epithelium doesn't have its own blood supply. So to prove my point, I want you to take a look at this picture. Can you see the yellow colored cells in there? Can you see that they are tightly adherent to each other? Okay, these cells are actually the cells of the epithelium. Okay, and I want you to refer to the area below the epithelium. Okay, it's actually labeled connective tissue. Okay, can you appreciate the presence of the red colored capillary in there? Okay, so that will only tell you that the connective tissue has its own blood supply and it's willing to share its blood supply to the epithelium. Okay, as long as the epithelium will also protect it from the hydrochloric acid in the stomach. So let's apply that one on an actual histology specimen. Okay, black arrow is actually pointing to the lining epithelium of the organ. As you can see, there's a simple columnar epithelium in there, and you can clearly see that the cells are tightly packed. Okay, and then I want you all to focus on the yellow star. The yellow star is actually situated on the connective tissue of that organ. Okay, can you appreciate the presence of a blood vessel above the yellow star? Okay, so that will actually tell you that the connective tissue is highly vascularized. So therefore, we can conclude that the connective tissue provides blood supply to the epithelium. I want you all to take a look at the photo. Okay, answer this question: what links the epithelium to the muscle? The answer is connective tissue. So that's the reason why it's called connective tissue, because it tends to connect the epithelium to the other tissues present in the organ. So for you to easily remember the things that you have learned from the previous slides, I want you to imagine the epithelium talking to the connective tissue, and then the epithelium would say, connective tissue, I will always be here to protect you. And then the connective tissue will respond by saying, don't worry epithelium, I will be here to keep you alive by providing you with oxygen. And then upon hearing them, the muscle tissue would say, sana all. And the third type of tissue that we can find in an organ is actually the muscle tissue, and it's actually incorporated in the organ to provide movement. And for us to feel that there's something wrong with the organs in our body, okay, so there must be the presence of the nervous tissue. So let's say, for example, in the case of the stomach, let's say there's erosion of the lining epithelium secondary to ulcer formation, okay. So for us to know that, the body will actually make us feel the pain, and the pain is because of the function of the nervous tissue. So let's try to recall how did we describe or define tissue in our previous discussion. We said that the tissue is made up of group of cells with interrelated functions. So let's try to take a look at this picture. As you can see, there's the presence of multiple cells in the picture, right? And these cells have interrelated function. But I want you all to appreciate that not all of the spaces in the tissue are occupied by cells. So what am I trying to tell you? There is more to tissue than just group of cells with interrelated function. Why? Because you can see their hair-like structures, which we call the fibers, okay, and empty spaces, okay, composed of chemicals, and those empty spaces are called ground substance, okay. So a tissue is actually made up of group of cells with interrelated function plus fibers plus ground substance. I want everybody to take a look at this picture, okay? The blue arrow is pointing to a cell, okay, which is one of the components of a tissue. And then the yellow arrow is pointing to the thread-like structure, okay, which is representing the fiber, which is also, again, one of the components of the tissue. And the red arrow is pointing to a empty space, but that empty space is actually made up of chemicals. And that empty space is actually called the ground substance. And I want everybody to understand, since the fiber and the ground substance are found outside the cell, they are referred to as extracellular matrix. Okay, so let's try to have a review. A tissue is made up of group of cells with interrelated function plus the extracellular matrix, which is further composed of fiber and ground substance. So again, the extracellular matrix is composed of what? Fibers and ground substance. So I want you to take a look at the animation. The blue colored structures there are representing the cellular component of the tissue, okay? And then also highlighted are the fibers. And the empty spaces that you can find between the fibers are actually the ground substance. So let's apply what you have learned from the previous discussion on this picture. The red arrows are actually pointing to the cellular components of the tissue, okay? The yellow arrow is pointing to the fiber component of the tissue, and the dark blue or black arrow is pointing to the ground substance. Ground substance and fiber will make up the extracellular matrix. Do you know that if only histology is taught using this kind of pictures, it would be easy for students to identify the components of the tissue? Okay, let's say, for example, if the cell would appear yellow in color, then you will have an idea it's an adipose cell or a fat cell, okay? Or if the cell appears to have a lot of dark granules, okay, then most likely you will identify the cell as mast cell, okay? If only histology is taught using these photos, okay? Okay, but the problem is, in reality, this is how this tissue will actually look like under the microscope. Let's proceed to the next slide. So this loose connective tissue will appear, move to the next slide, like this under the microscope. So if you only rely on the idea that anything yellow is adipose cell, anything with dark blue granules is mast cell, you will not most likely identify the cells in an actual tissue. Okay? But I promise you, at the end of this semester, you can identify that the predominating cells there are fibroblasts, the bigger fibers are collagen fiber, the thinner fibers are elastic fibers. But as of now, okay, it's enough that you can identify which is the cellular component of this tissue, okay, which is the fiber component of this tissue, and which is the ground substance, okay? But don't worry, we will be spending time to teach you how to identify the cells in the photo. I promise you that, okay? In your histology laboratory, you will be actually asked to view histology slides, okay? And you will be asked to view these slides under the microscope, okay? Shown on the left side of the screen is a human appendix, okay? And then the photo on the right side of the screen is showing you how the appendix will actually appear under the microscope. So I want everybody to imagine, you know how small a glass slide is, okay? So the next question is, how can you fit, okay, a large human organ into a glass slide so that you can study them under the microscope, okay? So in the laboratory, we do not get one slide and prepare another slide and place the appendix between them and crush the appendix using these slides, okay? We do not do that in the laboratory. Instead, so in histology laboratory or histopathology laboratory, we prepare the organ and we make a thin cut section of the organ. And the main purpose of preparing a thin section is that so that when we place this thin section on top of the slide, okay, the light coming from the light source of the microscope can actually traverse through the slide and the specimen so that the light can reach the eye of the observer. Okay, so I want you all to take note that what if, okay, we will make a thin cut section of this organ and you will look at it from the direction of the guy in the photo, okay? So let's say, for example, this organ is the appendix. We made a cross section of the appendix and we will actually look through it from the direction of the guy in the photo. We will most likely appreciate the cross section of the appendix and it will actually appear this way, okay? So again, it's very important in histology laboratory to prepare thin and translucent histological sections of the organs that we need to study so that we can clearly see them under the microscope. But the problem is before we can prepare a thin histological section of the organ that we need to study in histology, the organ has to undergo different tissue processing steps. Okay, so shown in the photo are the different steps that the organ has to undergo, okay, so that we can prepare the thin section. And it will always start with fixation. So let's try to discuss fixation first, okay? The first step will always be fixation, and the main purpose of the fixation is to preserve the organ and to prevent changes in the organ. Okay, for you to understand that, I want you all to take note that if you remove an organ from the body because you want to process it, you have actually cut it from its own blood supply. If there's no blood supply, then most likely the cells of that organ will not receive oxygen and the cells will start dying and the organ will begin decomposing or decaying. Okay, so let's say how important is fixation? So let's say, for example, so let's say we have a patient who came in because of right lower quadrant pain, and the patient is diagnosed to have appendicitis. And you know very well the best management for appendicitis is appendectomy or surgical removal of the appendix. So the patient will undergo the operation and the appendix will be surgically removed. Okay. Shown in the photos are two appendices, okay? The smaller one is a normal human appendix and the larger one is actually an inflamed appendix. So just try to imagine the difference between the normal appendix and an inflamed or infected appendix. So upon removal from the human body, the appendix should be immersed in formalin for fixation. And the main purpose of which is not to allow the organ to undergo decomposition or decay. Okay, so take a look at these pictures. On the left side of the screen, you have there an inflamed appendix that was preserved because you did the fixation step. And the appendix on the right side of the screen was not fixed after the surgery. So the organ undergone decomposition or decaying. Normally, when we view appendix in the laboratory and we want to diagnose whether the patient has acute appendicitis, we will actually shift the objectives of the microscope to a higher magnification, and we will try to look for, okay, so I want you to take a look at this. I want you to take a look at the first photo. That is how the appendix will look like under the scanner view, okay, or scanner objective of the microscope. but we will try to investigate the different areas of the appendix. Okay, shown on the right side of the screen is the high power magnification view of the appendix on one of the areas in the appendix. Can you appreciate that there's the presence of multiple neutrophils there? Okay, the cells that you see in there are actually neutrophils, and if you can recall from your anatomy, neutrophils are antibacterial. Okay, these are white blood cells that fight off bacteria. So if you have abundance of these neutrophils in your appendix, it would suggest that the appendix is having a current infection, and that will confirm, okay, the diagnosis of the doctor or the surgeon that the patient is really having acute appendicitis. Okay, but the problem is if the medtech, okay, forgot to fix the organ, or if the surgeon, after removing the appendix from the body of the patient, forgot to immerse the organ in the formalin or in the fixative agent, then most likely you will no longer appreciate the presence of these neutrophils because the appendix will undergo decomposition and decaying. So that's the importance of performing fixation. So there are different purposes for fixation. Number one, avoid tissue destruction by digestive enzymes. So let's say, for example, the organ is the small intestine. Okay, and you know very well that is where the enzymes from the pancreas are drained onto. Okay, so if the intestine is removed, okay, and if the enzymes are still there and the organ is not fixed, then these enzymes are not inactivated. The enzymes will start digesting the intestine, and you will no longer be able to appreciate the structures of the intestine. So that's what we mean by number one. Number two, the fixation step will actually terminate cell metabolism. And what's the purpose of terminating cell metabolism? So I'll just read this one and I'll explain this one on the next slide. Without oxygen, glycolysis. If I hope you can still recall from your favorite subject last semester. Glycolysis is actually the conversion of glucose to energy, right? Okay. If glycolysis will happen without oxygen because the organ was already cut off from its blood supply, okay, then the product will be lactic acid. So to best explain this one, I want you to proceed to the next slide. So I want you to take a look at the picture. When you have oxygen, glucose will be most likely converted to pyruvate, and pyruvate will enter the mitochondria to complete the Krebs cycle and the electron transport chain, and there would be generation of ATP molecules. Okay, but what if, okay, you look at the lower part of the photo, in the absence of oxygen, okay, the glucose now will be converted to lactic acid or lactate. Okay, from the word itself, lactic acid. So if the appendix was removed from the body, and the cells there will use up the glucose, the remaining glucose in the appendix, and then instead of producing energy, they are building up lactic acid. The lactic acid, okay, will start destroying the appendix. So that's the reason why we have to terminate cell metabolism. We should not allow the glycolysis to happen, or else the cells will start producing lactic acid, and the lactic acid will destroy the appendix. Okay, I hope I was able to explain that well. If you have any questions, you can simply ask me through our Telegram group chat. So that's the purpose of creating the Telegram group chat. You can chat me as late as 3 a.m. I'm still doing some stuff kasi until 3 a.m. So you can ask me questions. Okay? So for the information to be retained in your mind, I want you to imagine someone who's doing a workout. Okay? Every time we're doing workouts kasi, we're making the muscles contract. And when muscles contract, they actually impinge, okay, on their blood vessels. So, as you do the workout, okay, the muscles are contracting, the blood vessels are impinged, then most likely the muscles are not getting enough oxygen or blood supply, right? So there would be a buildup of lactic acid in the muscle. And the buildup of the lactic acid now will now be felt as the pamaol, okay? So every time you go to the gym, or every time you do the jogging and you go home and you feel pamaol, and your mother will ask you, "What's wrong with you, anak?" Or you will tell your mother, "Ma, lactic acid buildup." At least your mother will know that you're really studying. Diba, the main purpose of doing the tissue processing is for us to create a thin section. Okay, I want you to imagine a soft organ in the body. Okay, If you're going to cut a soft organ in the body and it's as soft as the gelatin, most likely the organ will move while you're doing the cutting, and you cannot actually make a perfect thin section. So one of the purpose of fixation is to actually somehow harden the organ so that it can facilitate now easy cutting or easy sectioning of the organ. Okay? So the third purpose of fixation is to harden the tissue by cross-linking or denaturing proteins so that it would be easy for the medtech to do the tissue sectioning later on. Okay? And number 4, the fixation also kills pathogenic microorganisms. Some of the specimens that are surgically removed from the body are infected. Okay? And to protect the people who are working in the laboratory, especially in histopath laboratory, the fixation has to kill the pathogenic microorganisms. And I want you all to take note, the most commonly used fixative in the laboratory is formalin. The next step to fixation is decalcification. Okay, there are organs that have calcium that makes them hard, and it would be difficult for the medtech to do the tissue sectioning. So for soft tissue, we need to harden them a little so that we can make the cutting. In cases of calcified tissue such as bones and teeth, okay, we need to decalcify them to somehow soften them so that we can easily cut them. Okay, so I want you all to take note, decalcification is only done, okay, in organs in the body that are calcified, and the examples are bones and teeth. Okay, and the most commonly used reagent for decalcification is nitric acid. So if I will ask you in the quiz, okay, what's the first thing that we should do if we receive bone in the histopath laboratory? The answer should be fixation. Everything should start with fixation. And the second step will be decalcification. But if the specimen is a soft tissue or soft organ such as the lungs, the heart, okay, we do first fixation, but there's no need for us to do decalcification. So I hope that's clear, ha? Bone, fixation, decalcification. Lung, fixation, skip the decalcification step. Decalcification is not only limited to bones and teeth. Okay, it can also be done to arteries, especially arteries with atherosclerotic plaque formation. Atherosclerotic plaque formation is actually caused by the deposition of the cholesterol on the walls of the lumen of the artery. So I want you to appreciate the yellow colored cholesterol deposited on the wall of the artery. And the presence of this cholesterol will actually trigger inflammation. And you will learn later on, especially in histopath, that whenever there is inflammation, there's also accompanying tissue destruction. And if there's tissue destruction, okay, then there would be deposition of calcium. So that area with the atherosclerotic plaque formation will somehow harden. Okay, and for us to sample that one, we need to somehow decalcify it also. So the next step to fixation or decalcification is dehydration. Okay, and dehydration means removal of water. So in this tissue processing step, we will be removing water from the tissue. And how do we do dehydration? You really have to take note of this. We do dehydration by successively dipping or bathing, okay, the tissue in a mixture of alcohol. Okay, and the most commonly used alcohol is actually ethanol. It's mentioned in the PPT, ethanol and water. Why do we have to use water? Because dehydration starts with using lower concentration of alcohol. So we can use 50% or 70%. Okay, and since when you say 50 or 70%, that's a diluted form of alcohol, so there's water in it. And I want you to take note, from 70, we will transfer the tissue to a higher concentration of alcohol. That could be 80 and 90. Okay? Then, until such time that the specimen will now be immersed on a pure or 100% absolute alcohol. Okay? So, again, dehydration is done by immersing, bathing the specimen from a lower concentration of alcohol to a higher concentration of alcohol until such time you're already dipping or immersing the specimen in 100% or absolute alcohol. And most of you would wonder, why do you have to immerse the specimen from a lower concentration of alcohol to a higher concentration of alcohol? That actually would take time, right? Okay, so to answer that question, because actually one of my Korean students in SPC before stood up during the class and asked me, why do we have to do that? It's kind of tasking, and it would require, longer processing time. So this is how I answered him. So I told him, Jihon, okay, you take a look at this grapefruit. Okay, the grapefruit on the right side of the screen was immersed immediately to 100% alcohol, while the grapefruit, okay, on the left side of the screen was immersed first on a lower concentration alcohol and was then transferred to higher concentrations of alcohol. So I asked the Korean student, okay, which among these steps, yung directly immersing the specimen to 100% alcohol or starting with the lower concentration of alcohol, will produce minimal tissue distortion. Okay, so the answer is you really have to start from a lower concentration of alcohol, then immerse the specimen to higher concentrations of alcohol, because you really want to preserve the morphology and the appearance of the organ as it was removed from the human body. So in dehydration, we remove water, we kick out water from the tissue, and the water will be replaced by alcohol. So the tissue now contains alcohol. Okay, the next tissue processing step is clearing. Okay, in clearing, so the next step to clearing is actually embedding. Okay, and embedding makes use of melted paraffin. That's why this procedure is usually done inside an oven, okay, set at 52 to 60 degrees Celsius, because that's the melting point of paraffin. The clear fluid, okay, that you can see in the picture to where the tissue is about to be immersed is actually melted paraffin. And since melted paraffin, okay, and the procedure is done at 52 to 60 degrees Celsius, that's actually a high temperature. So what will happen? Once the tissue is dipped into the paraffin, the xylene or the toluene will immediately become vapors, allowing now the melted paraffin to infiltrate the tissue. So in embedding, we remove the clearing and we replace it with paraffin. So that's basically the essence of doing embedding. So this tissue here in the picture will be immersed on that melted paraffin, and the melted paraffin will be allowed to cool down, forming now a solid block containing the tissue in the center. So again, in embedding, the tissue is placed in melted paraffin, and the melted paraffin is prepared in an oven at set at 52 to 60 degrees Celsius. With this temperature, the clearing agent will really uh, vaporized. Okay, so that's the explanation of the second bullet. So I know you have already understood the explanation, huh? So the paraffin now will be allowed to cool down, okay, so that you will now have the formation of a block with the tissue placed in the center. The problem with using melted paraffin, a lot of medtechs are prone to having what? Burn injuries. That's why in some laboratories, paraffin is already replaced by plastic resins. Okay, these plastic resins are fluid at room temperature, but once you try to place something on it, it will try to solidify immediately. Okay, so there's no need for you to do the procedure using an oven, and there's no chance that you will get burn injury. So this is an example of a plastic resin. As you can see, the materials that are placed on the plastic resin will most likely be preserved because the plastic resin has already solidified upon the addition of these materials. The problem with using plastic resin is that they are too expensive, and the laboratories in the Philippines cannot afford to use plastic resins, because most likely if they will use plastic resins, they have to increase the prices that they are charging on biopsies, and most likely some of the patients will not be able to afford it. So let's go back to the traditional paraffin. Okay, so take a look at this tissue block here. Okay, so you can see you have the hardened paraffin with the tissue in the center. So this is just a picture of the other samples that were processed in histopath laboratory. As you can see, you have there the tissues placed within the solidified paraffin. So this is embedding. Okay, this is embedding. So the next step to embedding is actually cutting. Can you appreciate in the picture that the tissue block contains excess paraffin? Okay, so cutting would involve the use of a knife, and the knife will be used to remove the excess paraffin. So that's how easy cutting is done. Okay, so again, after the tissue block has hardened, it is trimmed into a smaller block size, okay, by removing excess paraffin. The next step is actually sectioning, okay, and I will no longer read this one, okay. I will explain sectioning on the next slide. In sectioning, we're actually using an important instrument, and we call it as the microtome. So the instrument that you can see in front of you is the microtome. Can you appreciate the tissue, okay, that is placed within the tissue holder of the microtome? Okay, so that's the reason why we need to remove excess paraffin, so that the tissue block will actually fit into the tissue holder or block holder of the microtome. Okay, I want you to appreciate that below the tissue holder or the block holder, you have there the steel knife. Okay, and then I also want you to take note that there is that drive wheel in there. So as you rotate the drive wheel, what will happen is the tissue block or the block holder will actually go up and down, and it will come in contact with the knife. Okay, again, as you rotate the drive wheel, the block holder will actually go up and down, and the tissue block will come in contact with the stationary knife. Okay, so as the block holder will go up and down, okay, the tissue block will come in contact with the knife. So as you can see in this animation, as the drive wheel is rotated, okay, the block holder will come in contact with the knife. And you know what will happen? So as the block holder will go up and down, the tissue block will come in contact with the knife, and you will now have the production of the thin sections. So that's the reason why this procedure is called sectioning, okay? Because using the microtome, you can now have thin slices or thin sections of the organ that you want to study. And the main reason why you need to place the tissue on the melted paraffin is for us to have something that will hold the organ during the sectioning. Okay? So, I want you to recall dehydration. Diba in dehydration, we remove water. Okay? So, I told you in that certain PowerPoint slide that I will explain to you later on why do we have to do dehydration. Remember, in embedding, we immerse the tissue in melted paraffin. Okay? And if you can remember, when you were younger, you were playing with candles, and you're allowing the candles to drop, okay, yung melted portion of the candle to drop on the water, and you notice that the candle or the paraffin in the candle will never ever mix with water. It will just simply float on top of the water. So if the tissue has water and you immersed it into the melted paraffin, then most likely embedding will not be successful because the paraffin will not be able to infiltrate the tissue. So what we did was we remove water, we replace it with alcohol, and then the alcohol is replaced with xylene because xylene will readily what you call this vaporize in the presence of the melted paraffin. So that's the reason why we need to do dehydration. Okay? I hope I was able to explain that well, ha? But I'm more than willing to explain that to you. Just chat me through our Telegram group chat. So what we will do is, we will allow the thin sections to float on water, placed on the water bath, okay? Then we'll just try to scoop them out using the slide, okay? Remember, the sections that we created using the microtome are still transparent. So even if you have already placed them on top of the slide and view them under the microscope, you won't be able to appreciate the structures, the composition, the cells within the tissue because the tissues are still transparent. That's why after sectioning, after placing the section on the slide, it is very important that we should perform the next tissue processing step, which is staining. Okay? You need to impart color onto the tissue so that you can see the structures under the microscope. before we will proceed with the staining proper, we will first perform deparaffinization and rehydration. So from the name of the procedure, deparaffinization, meaning we're going to remove the paraffin from the thin sections. So how do we remove paraffin from the thin sections? We will use silene. I hope you can remember in the infiltration or embedding, silene is replaced by the paraffin inside the hot oven. In deparaffinization, we're going to remove paraffin using silene. So the thin section now is infiltrated with silene. And there's a need for us to remove silene. And how is that made possible? We're going to remove silene by using absolute alcohol. Okay, so the tissue section at this point will now be infiltrated by absolute alcohol. Later on, the tissue section will be immersed in 95% alcohol. So as you can see in dehydration, we're going to immerse the tissue in ascending concentration of alcohol. This time, we're going to remove silene by immersing the tissue into a decreasing concentration of alcohol. So after performing or immersing the tissue in 95% ethanol, expect that the tissue has alcohol. We are going to remove alcohol by immersing now the tissue section in distilled water. So we will allow water to go back to the tissue. I hope you can remember in the dehydration step, we removed water and replaced it with alcohol. This time, in rehydration, we are going to remove alcohol by using distilled water. What do you think is the purpose of rehydrating the tissue? Most of the stains that we are utilizing in histopath laboratory or histology laboratory are water soluble. So for the stains to impart color to the tissue must contain water. After performing deparaffinization and rehydration, we will now proceed with performing staining. Staining is important because the sections that we have prepared are still transparent. Staining is defined as the application of color to the tissue to highlight their structures. And take note, the two most commonly used stains in histology laboratory are hematoxylin and eosin. And these two are water soluble. That's why it's very important to rehydrate the tissue section. I want everybody to understand hematoxylin imparts blue or purple color. Okay? Hematoxylin imparts blue or purple color. And eosin, on the other hand, will impart red or pink color. Everybody listen. Hematoxylin is a basic dye. It's an alkaline dye. If you are an alkaline dye, okay, you're supposed to be attracted to the opposite. Okay, so hematoxylin, being a basic dye or an alkaline dye, will be attracted to portions in the cells that are acidic. Okay, I hope that will stick in your mind, ha? Hematoxylin, being a basic dye, will impart color to the acidic portions of the cell. Eosin, on the other hand, is an acidic dye. So therefore, it will impart color or it will stain the basic or alkaline portions of the cell. Okay. So I want you all to take a look at this picture. Okay. Just focus on one cell, the cell that is pointed with the blue arrow. Okay, the round structure that you can see inside the cell is the nucleus. Okay, and what do you notice? The nucleus is blue in color. So if you will be asked in the exam, what imparted color to the nucleus, the answer is hematoxylin. So let's try to analyze. Hematoxylin is a basic dye. Okay, and it's supposed to be attracted to acidic portions of the cell. Okay, and in the picture, it's very obvious that the hematoxylin was attracted to, or it has an affinity to, the nucleus of the cell. Why? What do we have in our nucleus? You've learned in your biochem, okay, in transcription, translation, discussion, that we have our DNA in our nucleus. And DNA stands for deoxyribonucleic acid. So therefore, our nucleus is acidic. And since it is acidic, it will be attracted to an alkaline dye. So that's why it retained the hematoxylin. Okay? Look at the cytoplasm of the cell. It's red in color. So most likely, the cytoplasm retained the eosin dye. So this is just a review of what you have learned on the previous slide. So I'll just give you 5 seconds, okay, to read the content of the PPT. 5, 4, 3, 2, 1. So let us try to define the terms basophilic and acidophilic. Okay, so I want you to recall the hematoxylin dye is a basic dye and it stains the acidic portion of the cell. Okay, if the portion of the cell retains the basic dye, okay, it's called basophilic. Okay, so in the case of the cell, the nucleus retained the hematoxylin, which is a basic dye, then the nucleus will be described as basophilic. Okay, why basophilic? From the name itself, basophilic. It has a love for basic dye. Okay, I hope you will not get confused. Hematoxylin is a basic dye and it stains the nucleus. Therefore, the nucleus is basophilic because it loves basic dye. Okay, if the structure is stained by eosin and eosin is an acidic dye, then that structure is called acidophilic. To make things more confusing, let's go back to the nucleus. Since the nucleus is stained by a basic dye, it's called basophilic. But in reality, the nucleus is acidic. Nucleus is acidic, therefore it is considered as a basophilic structure because it will attract basic dye. I hope it makes sense. I hope I was able to explain that well. Okay. So this one you really have to memorize by heart, okay? Because there's no way I can explain this one because this is just purely memorization, okay? But I only placed 4 important stains or 4 special stains in histology. I removed the other stains because they are best discussed in histopath, okay? And some of them are best discussed in bacteriology in 3rd year. Okay, for now, let's just settle with these 4. Fuel-Gen reaction for DNA, periodic acid shift for carbohydrates, okay, Sudan black for lipids, and silver stain for reticular fibers. So I really want you to memorize these 4 by heart, okay? So, how long would it take for the tissue to be fixed, dehydrated, cleared, embedded, sectioned, okay, mounted, and stained and read by the pathologist? In the Philippine laboratory setup, the tissue would require at least 48 to 72 hours tissue processing time. Okay, so meaning if the appendix of the patient is removed, okay, then it is biopsy, then expect that the patient will have the result after 2 to 3 days. Okay, that's how long the tissue processing would take. We have special cases such as this one. So I want you all to listen. So let's say we have a patient who has a colon cancer and the cancer is located on his transverse colon. Okay, so the surgical plan is to remove that portion of the transverse colon with the mass or the cancer, okay, and anastomose or connect or surgically linked, okay, the remaining portions of the transverse colon. But before you anastomose the remaining portions of the transverse colon, you really have, or as the surgeon, you really have to be sure that the remaining portions, okay, the ones that are left inside the human body should no longer contain cancer cells, or else, okay, you should expect the cancer to reoccur or recur, okay. So what will the surgeon do is, okay, before the surgeon will anastomose, okay, the 2 portions of the or the remaining portions of the transverse colon, the surgeon will have to send the mass, okay, or the cancer tissue to the laboratory. So what will happen is the patient will undergo surgery, the surgeon will remove the portion of the transverse colon with the mass, okay, and this portion of the mass, the resected portion of the transverse colon, will be sent to the laboratory. And the surgeon will not immediately proceed to anastomosis. Why? The surgeon has to make sure, okay, that the remaining portions of the transverse colon in the body of the patient are cancer free. Okay, so what will happen now to the resected transverse colon mass in the laboratory? So the pathologist will actually sample or do a biopsy of the edges or the borders of the last but not the least among the tissue processing steps is mounting. In mounting, we would be placing cover slip on top of the stained specimen. And the main purpose of which is to preserve the specimen, to prevent washing off of stain, to prevent contamination, and to provide support. Before the glass slide is placed on top of the stained specimen, we need to place mounting media such as glycerin or resin. And the main purpose of these chemicals is to make the cover slip stick into the specimen and glass slide. Then, you can now view the stained specimen under the microscope. So the shortcut method I've been telling is the frozen section. Okay? This time, you do not anymore immerse the specimen in formalin and then dehydrate the specimen and then do the clearing procedure and embed the specimen. This time, okay, you will just do rapid freezing, okay, using compressed carbon dioxide. I hope you can still remember in some sci-fi movies yung may gas, okay, and then if the actor will come in contact with the gas, the actor will actually freeze, okay. That's actually compressed carbon dioxide, okay. And we will be using an important instrument here. It's called cryostat, okay. And how does a cryostat look like? Let's proceed to the next slide. It actually looks like a huge refrigerator, okay. So since compressed carbon dioxide needs to be maintained at a cold temperature, so the entire procedure should be done within a cryostat, okay. And what do you notice? You have there a built-in microtome within the cryostat, so that you will, after freezing the tissue, you will immediately do the sectioning. So you are actually you are actually skipping the other procedures, yung dehydration, clearing, embedding. From fixation using the compressed carbon dioxide, you can immediately go to sectioning inside by using the cryostat. So you have a microtome inside a refrigerator-like apparatus, and the name of that refrigerator-like apparatus is the cryostat. So frozen section is a rapid method. Okay, it's routinely done in hospital to study specimens during surgery. Okay, and since the tissue is not exposed to many chemicals, unlike in the routine tissue processing, then most likely lipids, enzymes are preserved using this method. Thank you for spending time to listen to my audio lecture. I hope you learn a lot of things. If you have questions, please feel free to send me a direct message in Telegram or in Twitter or in Facebook. Okay, then I will still be uploading another PPT. Okay, so the quiz this Saturday will include this lecture and the second PPT that I will be sharing to the group. Okay, so good luck everyone. Resected colon. Okay, and the main purpose of doing which is to look for the presence of cancer cells. If the pathologist will not see any presence of cancer cell on both edges of the transverse colon or the resected colon, then it will give the pathologist and the surgeon the message that the remaining portions of the transverse colon in the body of the patient are cancer-free. Therefore, the surgeon can proceed with the anastomosing. But if the pathologist will find presence of cancer cells, okay, on one of the edges, okay, the pathologist will call the surgeon and advise the surgeon to do more resection, okay, and that is to prevent the recurrence of the cancer in the patient. Okay, so what is happening here is that the surgery will not end not unless the pathologist will release the result, right? And remember, the routine tissue processing would take 3 to 4 days. And we cannot make the surgeon and the patient wait for 3 to 4 days before the patient will be closed up. Most likely, the patient will wake up in the middle of the surgery and say, Dok, kuman na, Dok, mutanaw, pabiya kong paborito na ako na salida sa Netflix, Dok. Na, we cannot afford for that to happen, no? We cannot allow that one to happen, I mean. So, there must be a shortcut method wherein the pathologist can simply get a portion of the resected mass, okay, and immediately make a thin cut section, okay, and view the specimen under the microscope. The shortcut method that I've been telling you is we will actually remove alcohol. Okay. In dehydration, we remove water, we replace it with alcohol. In clearing, we will now remove the alcohol and replace it with the clearing agent. And I want you to take note, there are two clearing agents that are popularly known, xylene and toluene. And xylene is the most commonly used clearing agents in the laboratory. Okay? So again, dehydration, remove water, replace it with alcohol. In clearing, remove alcohol, replace it with xylene. And I want you to take note that clearing agents, okay, are usually highly volatile. So meaning if the tissue with the xylene will be exposed to a high temperature, the xylene will immediately evaporate, leaving behind spaces within the organ. The word tissue, you should expect to see what? Group of cells with interrelated functions. Okay? In the nervous tissue, these cells are categorized into neurons and neuroglia. Okay? The cell that you can find in the center is actually the neuron. You know very well the neurons function to create nerve impulse. So that, let's say for example, your brain wants one of your muscle to move, okay? For the brain to make that muscle move, the neurons, okay, will generate nerve impulses. Okay? I want you all to take a look at the cell labeled as microglia. Okay? It's one of the neuroglia in the nervous tissue. This microglia is actually a phagocytic cell. It's actually there to what? Eat up bacteria that will try to kill the neurons. So you see, the function of the the microglia, the function of the microglia is there to what? To support, to keep the neurons alive. I want you to take a look at the cell that is labeled as astrocytes. Okay, it's named such because it's star shaped. Okay, because when you refer to the term astro, it means kasi star. Okay, what do you notice? Some of the processes of the astrocytes are reaching to the neurons, while the other cytoplasmic processes are wrapping around the capillary. Okay, do you know how important astrocytes are? Okay, they actually limit the entry of substances from the capillary or blood to the nervous system. Okay, because I want everybody to understand there are toxic substances that you can find in the blood that once they come in contact with the neurons, they can actually kill the neurons. And what will limit their entry or diffusion, okay, out from the capillary is actually the presence of the astrocyte. As you can see, the function of the astrocyte is to keep the neurons alive. The function of the microglia is to protect the neuron from the invading microorganisms so that the neurons can actually continue generate nerve impulses. So that's what we mean now by a tissue, group of cells with interrelated function.